In this video, I want to talk a little bit about the physiology of reproduction in women, including formation of the oocyte, the female gamete. This diagram shows you the events of oogenesis. Oogenesis is the production of the female gamete, production of the egg. Notice that this diagram is divided into two columns. On the left here, you have the events of meiosis. So this is what's actually happening to develop an oocyte. On the right, you have development of the follicle. Remember that the follicle is the little pocket within the ovary that an oocyte sits in. And as the oocyte goes through development, the follicle goes through different stages as well, preparing to nourish and give that oocyte the best chance of developing a healthy pregnancy. So oogenesis begins with the oogonium. The oogonium is the egg stem cell, just like the spermatogonium is the sperm stem cell. So the oogonium undergoes mitosis, and then one daughter cell goes into development to become an oocyte. The other daughter cell, we know this because this is a stem cell, the other daughter cell remains as an oogonium. That's the definition of a stem cell, is that it replaces itself. Now that primary oocyte, we're still talking about a diploid number of chromosomes. Formation of the primary oocyte, this happens before birth. The primary oocyte basically pauses. It progresses to prophase 1 of meiosis and then just sits there, inactive, through infancy and childhood. The follicle at this point is known as a primordial follicle with the primary oocyte. Now once the girl enters into puberty, each month, a few oocytes begin to mature and start to progress through meiosis. Okay, so it goes through the first meiotic division, goes through meiosis 1, and then this meiosis 1 is a little bit different in oocytes than it is in spermatocytes, because at the end of meiosis 1 in spermatocytes, you get 2 functional cells, each of which goes into meiosis 2. At the end of meiosis 1 in oogenesis, you actually only get 1 functional cell. The second set of DNA, so the set of homologous chromosomes, are basically kicked out into a structure called a spindle and released as what's called a polar body. So instead of producing 2 functional cells, you produce 1 functional cell that can go into meiosis 2, and a polar body that may go into meiosis 2, but often doesn't. So the functional cell, the secondary oocyte, progresses into meiosis 2, but then it reaches metaphase 2 and pauses. And if this cell is released during ovulation, it's released in metaphase 2. It only completes meiosis 2 if fertilization occurs. The entry of a sperm, the penetration of a sperm cell into this secondary oocyte is what triggers completion of meiosis 2. And again, at the end of meiosis 2, you get another polar body formed that's released and not used, can't be fertilized. It's not functional. So one of the differences between oogenesis and spermatogenesis is that at the end of oogenesis, you get only 1 functional gamete. The other 3 are all basically eliminated, phagocytosed, broken back down, released. Also, kind of a unique characteristic of meiosis in oogenesis is that meiosis is only completed if fertilization occurs. If fertilization doesn't occur, if there's no sperm cell around to fertilize that egg, then the secondary oocyte is just shunted out. It's just released from the uterus, leaves the body. Now, while all this is happening, you have development of the follicle as well. The primordial follicle with the primary oocyte is present at birth and through infancy and childhood. During once puberty hits, as the oocyte matures, the primordial follicle becomes the primary follicle, secondary follicle, the vesicular follicle or Graafian follicle, sometimes called tertiary follicle, and then ovulation, the release of the secondary oocyte, is basically a rupturing of the follicle. Then after ovulation, that ruptured follicle becomes a structure called a corpus luteum, and I want to show you this progression in follicle development and talk about it in a little bit more detail. This slide shows the different stages in follicle development. It's a bit misleading because these stages, they're separated in time. They aren't separated in place. So a single primordial follicle can become a primary follicle, a secondary follicle, a Graafian follicle, and a corpus luteum. It's not a progression through the ovary. It stays in one place. This is just a convenience for illustration, showing it in separate locations. So here we've got the primordial follicles, which are quiescent with the primary oocyte. They begin to be activated to primary follicles at puberty, and they're continuously activated during puberty. The mechanism that sort of picks, quote-unquote, picks a primordial follicle each month to develop it into a primary follicle is really unknown. There's probably some local factors in the ovary, some local conditions in the ovary that say this primordial follicle is going to become a primary follicle. And it's probably mostly just random. Several primary follicles form, but only a few of the primary follicles progress to secondary follicles. So if an activated follicle doesn't mature into a secondary follicle and a Graafian follicle, it degenerates. And the process of follicular degeneration is just called atresia. So if a follicle doesn't mature, it undergoes atresia. That's an E. As the follicle matures, the cells in the wall of the follicle enlarge, divide until they become a thick layer of granulosa cells surrounding the oocyte. So this is the granulosa cells in a secondary follicle. About 8 to 10 days into a woman's monthly cycle, she only has one secondary follicle with separation of the inner and outer layers. By about 2 weeks into the cycle, days 10 to 14, the secondary follicle matures to a Graafian follicle. And in a Graafian follicle, you have the thick kind of curtain of granulosa cells. You have the oocyte in there, again surrounded by granulosa cells. But you also have a fluid-filled chamber within the follicle, and this chamber is the antrum. By day 14, the granulosa cells detach, and you actually have the oocyte, and it's sort of quart of granulosa cells floating in the fluid of the antrum. Here's a different image of the Graafian or tertiary follicle. You have the oocyte here with its big distinct nucleus. You have the zona pellucida, which is just this clear layer, almost like a soft shell around the oocyte. Then you have the granulosa cells, you have the fluid-filled antrum, and then you have more granulosa cells in the wall of the follicle. And there's actually a nice little primordial follicle right next door with the oocyte in it, so you can kind of compare the two. Now, the primary oocytes in the Graafian follicle are suspended in prophase 1 of meiosis up to this point. In the monthly cycle, the levels of luteinizing hormone begin to rise, and it's that rise in luteinizing hormone that stimulates the oocyte to complete meiosis 1, forming the polar body. The secondary oocyte, the one functional daughter cell, proceeds into metaphase 2 at this point, and then pauses again. At about day 14 of the menstrual cycle, there's a surge in luteinizing hormone. So the levels of luteinizing hormone, if you look over time in the cycle, they're fairly steady, and then they increase and then surge into a sharp peak. And this surge happens at about day 14 in the menstrual cycle. And the surge in luteinizing hormone weakens the wall of the ovary, so that that fluid pressure that's been building up in the antrum pushes through the wall of the follicle and releases the oocyte. So now the oocyte's released. This is in actual microscopic image of ovulation. You can see the oocyte in there and the ruptured follicle wall. At this point, the oocyte ideally gets swept into the uterine tube. Remember, the fimbriae kind of gently brush the ovary and provide a guide for bringing that oocyte into the uterine tube. Some women actually experience pain at the point of ovulation. There's a term for it from the German called Mittelschmerz, which just basically means middle pain, and it's not really well understood. It's known that some women actually feel it. It may be from the stretching of the ovary, it may be from irritation of the peritoneum as you have fluid released from the antrum. It's not really well understood, but some women can tell exactly when they ovulate. Now, we've been saying that we have one follicle that matures and one oocyte that's released during ovulation, but in a small proportion of ovulation events, about 1 to 2% of ovulations release multiple oocytes. So you got more than one oocyte maturing, more than one follicle maturing, and when you have multiple oocytes being released, if they all, usually not more than both, but if they get fertilized, you can get multiple births, fraternal twins. After ovulation, the granulosa cells that are left behind in the wall of the follicle form a structure called the corpus luteum. And that's what you're seeing here. This is actually a really large one. It's taking up a huge chunk of the ovary. They're not always this big, but that's the corpus luteum. And the corpus luteum is an endocrine structure. It secretes hormones, mostly progesterone and some estrogen. Progesterone stimulates maturation of the uterine lining and the secretions of the uterine glands. So these hormones that are released by the corpus luteum basically prepare the uterus for implantation of an embryo. So it's basically saying like, okay, we got an oocyte out there, let's get ready, we might have a pregnancy coming. If there's no pregnancy, the corpus luteum deteriorates after about 10 days. So in the absence of pregnancy, the corpus luteum becomes the corpus albicans, which is basically just scar tissue. It's called Corpus Albicans, means basically the white body, just like Corpus Luteum means the yellow body. And it's white because it's just non-functional scar tissue. So if you are experienced at examining ovaries, you can actually look at an ovary, look for these little structures of Corpus Albicans, and determine how many times that ovary has ovulated, because you can count the scars, the ovulation scars. If there is pregnancy, the corpus luteum continues secreting hormones throughout the first trimester, basically until the placenta takes over. If you remember from the previous video, the reproductive anatomy video, we talked about how the placenta doesn't form until about isn't fully formed until about 10 to 12 weeks after conception. The placenta is also an endocrine organ as well as providing nutrients to the fetus. And the placenta's endocrine function isn't fully developed until about 3 months into pregnancy as well. Now, there's a hormone that's released during pregnancy that keeps the corpus luteum active. You may have heard of it. It's called human chorionic gonadotropin, or HCG. That's the hormone that keeps the corpus luteum active if pregnancy occurs. If there's no pregnancy, there's no HCG, and the corpus luteum deteriorates and becomes a Corpus Albicans. You may have heard of HCG because it's the same hormone that's detected by pregnancy tests. So if you or someone you know has ever had to pee on a stick to determine if they're pregnant, what that test is looking for is the presence of HCG. HCG is actually produced by the embryo itself. So it's basically that fertilized embryo sending out a message that it's there and it needs to be supported. Okay, so there are a number of hormones that are involved in regulating the female reproductive cycle. I want to first talk about hormonal regulation of oogenesis, and then we'll get into the menstrual cycle, which is the preparation of the uterus for pregnancy, and reversing that preparation if pregnancy doesn't happen. So the main hormones that are involved in hormonal regulation of oogenesis include gonadotropin releasing hormone, follicle stimulating hormone, luteinizing hormone, and progesterone and estrogen. And I'd say luteal here, that just means released by the corpus luteum. This is a visual representation of the interactions of hormones and the regulation of hormones in the female reproductive system. You have the solid lines, in this case, represent a stimulatory interaction, so positive interaction. The dashed lines represent an inhibitory reaction, so negative feedback mechanisms. Gonadotropin releasing hormone, just like it is in males, it's released from the hypothalamus, and it triggers the release of FSH, follicle stimulating hormone, and luteinizing hormone from the anterior pituitary. And so that's the same as happens in males. However, the effects of follicle stimulating hormone and luteinizing hormone are different in females. Now, the names of these hormones are given for what they do in females, so you can kind of keep track of what they do based just on the names if you're talking about female reproduction. Follicle stimulating hormone stimulates follicle development, as you might guess. Luteinizing hormone, you get this surge in secretion around day 14 of the menstrual cycle. And that surge triggers completion of meiosis 1 by the primary oocyte, and it triggers ovulation. So that high level of luteinizing hormone triggers oocyte development and release. It also triggers the formation of the corpus luteum. That's why it's called the luteinizing hormone. Now, once the corpus luteum is formed, it secretes progesterone, some estrogens, and a small amount of inhibin. And these trigger a negative feedback mechanism. So these progesterone, estrogens, and inhibin from the corpus luteum, they suppress the gonadotropin releasing hormone release. They suppress luteinizing hormone and FSH release. So they suppress GnRH and they suppress LH and FSH. And it's that decline in luteinizing hormone that causes the corpus luteum to deteriorate. So it's a true negative feedback loop. Decline in the luteinizing hormone, the corpus luteum deteriorates, becomes scar tissue. That removes the negative feedback. And then once the corpus luteum has deteriorated and the negative feedback loop is gone, the release of GnRH starts to build again, and FSH and LH come back up. So that's where you get the cyclical nature. You get a positive feedback loop through here, and then negative feedback And then when this negative feedback is removed, you get the positive feedback coming through again. That's why you get a constantly renewing cycle. HCG, human chorionic gonadotropin, from a fertilized embryo basically kind of mimics luteinizing hormone. So since luteinizing hormone supports the development of corpus luteum, if you have HCG kind of acting a little bit like luteinizing hormone, it sustains the corpus luteum even though the mother's levels of luteinizing hormone are coming down. This negative feedback mechanism is actually the beginning of hormonal birth control for women. Since the progesterone and estrogens suppress gonadotropin releasing hormone and suppress the FSH and LH, hormonal birth control usually contains a combination of progesterone and estrogens, although there are formulations of the pill that are just the progesterone only. And so by having a continuous low dose of progesterone and estrogens, it keeps up the suppression of gonadotropin releasing hormone. And if gonadotropin releasing hormone is continuously suppressed by progesterone and estrogens coming in pill form, then there's never that LH surge, there's never ovulation. So that's how the hormonal birth control basically takes advantage of this negative feedback loop to suppress ovulation. Although it also has other mechanisms that work to release contraception, such as thickening the mucus in the vagina so that sperm can't pass through the cervix and enter into the uterus and uterine tube. With this diagram, we're starting to look at the menstrual cycle, the monthly cycle of ovarian development and uterine development that happens as part of a woman's reproduction. So you have gonadotropic hormone levels up here, the follicle stages that they correspond to, so follicle development in the first half of the cycle, corpus luteum formation and deterioration in the second half of the cycle. This also shows how these correspond to ovarian hormone levels. It all really starts with GnRH here, and GnRH is released in small pulses at about 60 to 90 minute intervals early in the reproductive cycle. This is also the rate at which GnRH is released in adult males. So a small pulse of GnRH about every 60 to 90 minutes. Now, this pulse frequency increases to every 30 to 60 minutes by mid-cycle. So about days 10 to 14, the levels of GnRH in the blood rise because you're getting a small amount of GnRH released every half hour to an hour. And it's the rising ovarian estrogen levels, actually, that triggers this increase in GnRH frequency. By about day 14, the GnRH pulse rate is every 30 minutes, and this high pulse rate triggers the release of luteinizing hormone from the anterior pituitary. That's when you get this surge in luteinizing hormone that triggers completion of meiosis 1 and rupture of the follicle wall. After this point, you get that negative feedback coming from the progesterone and estrogens of the corpus luteum, which inhibits the GnRH pulse rate, bringing the GnRH levels in the blood back down. So that's what's happening in the ovaries. Now let's talk about what's happening in the uterus on the same time frame. Menstruation is the shedding of the uterine wall, the shedding of the stratum functionalis, of the endometrium. And there is a cycle of growth, the proliferative phase, and production, the secretory phase, and then loss, the menstrual phase. That the uterine wall, that the endometrium goes through each month. The menstrual phase is when the functional endometrium is actually being shed And this is triggered by a decline in estrogens and progesterone. As those levels fall, there's not as much support to the endometrium. The blood vessels in the endometrium actually start to sort of kink, and so the endometrium isn't getting a sufficient amount of nutrients and oxygen, and the tissue starts to die and gets released. In the proliferative phase, the endometrium is rebuilding itself. The stratum functionalis is rebuilding itself. And this is stimulated by rising estrogen levels. As estrogen levels come back up, new blood vessels start to grow and start to fuel the development, the proliferation of cells in the endometrium. In the secretory phase, the endometrium is basically getting ready for implantation, and it's called the secretory phase because the uterine glands are functional. They're secreting glycogen into the lumen, preparing for an embryo to implant. And this is stimulated by progesterone released from the corpus luteum. The onset of menstruation is called menarche, so that's the first menstruation. And it varies oftentimes around about 12 and a half, but there's actually been some concern lately that girls are starting to go into menarche earlier for unknown reasons. It may have to do with hormones in the environment, it may have to do with dietary changes. There's interest in it, but not a lot of answers yet. And so that's when menstruation often occurs. That's when the first menstrual cycle begins. Menstruation continues until approximately the late 40s, early 50s. Menopause. So menopause is when menstruation stops. Just the estrogen levels decline, and the uterine wall doesn't rebuild itself anymore. This is normal. It's normal to have an onset of menstruation. It's normal to go through menopause in later middle age. It is possible to have an abnormal disruption of menstruation as well, and that's called amenorrhea. This can occur with severe physical stress, severe emotional stress. It can occur with anorexia. It's thought that the level of fat on a woman's body is a signal for menstruation, a signal for reproductive preparation, which in a way makes sense if you think about it. Someone who is starving and doesn't have enough nutrition, isn't getting enough nutrients to support themselves, they don't want to get pregnant and try to be supporting a developing baby. So in a way it makes sense to have the level of fat reserves on the body as almost a signal that, like, it's okay, you've got enough food, you can have a baby now. Conditions like anorexia, where there's a very, very low level of body fat, or even with like girls that may not be anorexic, but that may be extremely, maybe athletes in extreme physical training, they often have very low body fat and oftentimes develop amenorrhea, stop having their period. These images just show you differences in the way the uterine wall appears under the microscope at the different stages of the uterine cycle. So here you're looking at menstruation. You have the kind of dead, loosened stratum functionalis that's starting to separate from the stratum basalis. Here you have, in the proliferative phase, you have very healthy functional zone with well-developed uterine glands. And during the secretory phase, the uterine glands are fully developed. They're throughout the functional zone, and the functional zone itself is pretty darn big, is much deeper, much thicker than it was during the proliferative phase or during menstruation. This is the bottom half of that diagram I showed you earlier. If you take the ovarian cycle diagram and combine it with this, you can see exactly how the ovarian cycle corresponds to the stages of the uterine cycle. So here we're looking at fluctuation in the ovarian hormone levels, the increase in estrogens by about day 14, and then the increase in progesterone as the corpus luteum forms. And these are the stages of the uterine wall that these hormone levels correspond to. So in the menstrual phase, you have low levels of estrogen, very low levels of progesterone, so the endometrium is not supported and it dies off. In the proliferative phase, you have rising estrogen levels triggering regrowth of the functional zone, the functional stratum. In the secretory phase, you have high levels of progesterone, maintaining completing maturation of the uterine glands and maintaining the endometrium. These changes also correspond to body temperature, and sometimes in the process of trying to become pregnant, women will monitor their body temperature to figure out when they're ovulating, to figure out when the best time to get inseminated is. And there's actually a rise in body temperature that happens after ovulation associated with that rise in progesterone. And so that rise in body temperature is a signal that ovulation has occurred. And there's an egg there ready to be fertilized. So after studying this video, together with the female reproductive anatomy video, you should be able to describe the process of oogenesis and compare it to spermatogenesis. You should also be able to describe the stages of follicle development and the stages of menstruation, or to be more precise, the stages of the uterine cycle. You should be able to relate these to each other as well as relating them to the stages of oogenesis. You should also be able to describe the role of GnRH, FSH, LH, estrogens, and progesterone in female reproduction, and talk about the corpus luteum, why that's important.